8.5.4 Mitochondria. Mitochondria, singular mitochondrion, unless specifically stained, are not easily visible under the microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cells. In terms of shape and size also, considerable degree of variability is observed. Typically it is sausage shaped or cylindrical, having a diameter of 0.2 1.2 μm, average 0.5 μm, and length 1.0 to 4.1 μm. Each mitochondrion is a double membrane bound structure with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments, that is the outer compartment and the inner compartment. The inner compartment is filled with a dense homogeneous substance called the matrix. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of infolding called the cristae, singular crista, towards the matrix, figure 8.7. The cristae increase the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Mitochondria are the site of aerobic respiration. They produce cellular energy in the form of ATP. Hence, they are called powerhouse of the cell. The matrix also possesses single circular DNA molecule, a few RNA molecule, ribosomes 70S, and the components required for the synthesis of protein. The mitochondria divide by fission. 8.5.5 Plastids. Plastids are found in all plant cells and in iguanoids. These are easily observed under the microscope as they are large. They bear some specific pigments, thus imparting specific colors to the plants. Based on the type of pigments, plastids can be classified into chloroplast, chromoplast, and leucoplast. The chloroplast contain chlorophyll and carotenoids pigments which are responsible for the trapping light energy essential for photosynthesis. In the chromoplast, fat-soluble carotenoid pigment like carotene, xanthophylls and other are present. These gives the part of the plant yellow, orange or red color. The leucoplasts are the colorless plastids of varied shape and size which store nutrients. Amyloplast store carbohydrate, starch, example potato. Aleuplast store oil and fats, whereas the aleuroplast store protein. Majority of the chloroplast of the green plants are found in the mesophyll cells of the leaves. These are lens-shaped, oval, spherical, discoid or even ribbon-like. Organelle having variable length 5 to 10 μm and width 2 to 4 μm. Their number varies from 1 per cell of the Chlamydomonas, a green alga, to 20 to 40 per cell in the mesophyll. Like mitochondria, the chloroplasts are also double membrane bound. Of the two, the inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called stroma. A number of organized flattened membranous sacs called the thylakoid are present in the stroma, figure 8.8. Thylakoid are arranged in stack like the peels of coins called grana, singular granum, or the intergranal thylakoid. In addition, there are flat membranous tubules called the stroma lamellae connecting the thylakoid of the different grana. The membrane of the thylakoid enclose a space called a lumen. The stroma of the chloroplast contain enzyme required for the synthesis of carbohydrate and protein. It also contains small double-stranded circular DNA molecules and ribosomes. Chlorophyll pigments are present in the thylakoid. The ribosomes of the chloroplast are smaller 70s than the cytoplasmic ribosomes 80s. 8.5.6 Ribosomes. Ribosomes are the granular structure first observed under the electron microscope as dense particle by George Plaidy 1953. They are composed of ribonucleic acid (RNA) and protein and are not surrounded by any membrane. The eukaryotic ribosomes are 80s while the prokaryotic ribosomes are 70s. Each ribosome has two subunits, larger and smaller subunit. Figure 8.9. The two subunits of ATS ribosomes are 60S and 40S, while that of 70S ribosomes are 50S and 30S. Here, S. Severtberg's unit stands for the sedimentation coefficient. It is indirectly a measure of density and size. Both 70S and 80S ribosomes are composed of two subunits. 8.5.7 Cytoskeleton, a elaborate network of filamentous proteinous structure consisting of microtubules, microfilaments and intermediate filament present in the cytoplasm is collectively referred to as cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. 8.5.8 Cilia and Flagella. Cilia singular xylem and flagella singular flagellum are hair-like overgrowth of the cell membrane. Cilia are small structures which work like oars causing the movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for the cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that of the eukaryotic flagella. The electron microscopic study of a coelom or the flagellum shows that they are covered with plasma membrane. Their core, called the oxyneum, possesses a number of microtubules running parallel to the long axis. The oxyneum usually has 9 doublet of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of oxynemal microtubules is referred to as the 9 plus 2 array. Figure 8.10. The central tubules are connected by bridges and is also enclosed by a central sheet which is connected to one of the tubules of each peripheral doublet by a radial spoke. Thus, there are 9 radial spokes. The peripheral doublet are also interconnected by linker. Both the coelom and flagellum emerge from centriole-like structure called the basal bodies.